Good afternoon. My name is Quelle. I'm a professor of neuroscience and pharmacology at the University of Iowa, and I am also a leader of the Iowa Neuroendocrine Tumors 4. I want to thank the organizers for the invitation to speak today about what's new in neuroendocrine tumor research. I have no disclosures. So we all know that neuroendocrine tumors arise throughout the body, and my talk today is going to focus on pancreatic neuroendocrine tumors, particularly the grades 1 through 3, which are well differentiated and represent the majority of PNETs that are seen in patients. For those tumors, treatments have expanded beyond surgery and somatostatin analogs to include PRRT and other targeted therapies like everolimus. Those newer treatment options are due to a greater understanding of PNET biology and molecular phenotype, as shown in this review diagram that highlights a variety of activated or inhibited pathways that are seen in patient tumors. And the more that we understand about the biology, the better we are at developing new therapies that are specifically targeted against PNETs, like those that target activated mTOR kinase, or those that are targeted against cell surface receptors, the somatostatin tumor receptors. Unfortunately, we know that patients with unresectable disease will inevitably progress, and this requires new targets or combination therapies that will suppress and ideally cause regression of treatment-resistant tumors. This really demands deeper insights into the disease through additional studies of patient tumors, which I won't be talking about today, as well as analyses of PNET models. And those include cell lines, patient-derived spheroids and organoids, and animal tumor models. All of those are really important at facilitating in vitro testing and in vivo testing of new treatments that will guide our future clinical trials. So in today's talk, I want to give you a flavor of different types of neuroendocrine tumor research using models that have increasing complexity. So we're going to start with some studies in immortalized patient-derived tumor cell lines. And then I'll talk briefly about some studies using short-lived patient-derived spheroids and organoids. These are unlike tumor cell lines because they're three-dimensional and they have many different cell types, not just tumor cells. And then I'll conclude with studies that are being done in mouse PNET models in vivo. So BON and QGP1 cells have been the workhorses in the field to assess pancreatic neuroendocrine tumor biology and drug sensitivities, as shown in this response curve to the histone deacetylase or HDAC inhibitor, mositinostat, where you can see that increasing amount of drug leads to killing of the cells. Now notably, histone deacetylase inhibitors are really important because they alter gene expression in the tumor cells. And several groups have found that they increase the levels and expression of somatostatin receptors on the surface. This is really exciting because HDAC inhibitors could improve patient responses to PRRT, especially for those patients whose tumors are negative for the receptors, and we need to find ways to bring those receptors up so that we can target them. So this is, of course, of high relevance because somatostatin analogs are so useful in neuroendocrine tumor diagnostic imaging and therapy. But their use has been limited by the fact that not all neuroendocrine tumors express somatostatin receptors, or the receptor can be downregulated by various treatments. So others have discovered a different way to increase the level of somatostatin receptors on tumor cells. In this case, they were targeting a pathway in which the protein disheveled is upregulated, and that actually downregulates the expression of the receptors. So what they did in cells was they silenced the disheveled protein, and you can see beautiful upregulation in this pinkish orange color of the somatostatin receptors on the cell surface. So they then tested a variety of different drugs to see whether those would upregulate the receptor, and they found that they did. So the exciting thing is that these drugs could be useful, just like histone deacetylase inhibitors, for improving somatostatin and PRRT therapies. Now, we also use PNET cells to assess putative drivers of disease, as shown here for an oncogenic protein, Rabl6A, that my laboratory studies. Now, we've silenced its expression in cell lines and showed that those cells now have much lower ability to proliferate and grow. We've also examined the effect of Rabl6A overexpression, and we did this by injecting cells that overexpress the protein into mice. And over time, tumors will form. And we compared the overexpression of Rabl6A to a control called GFP. And you can see by the size of these tumors that overexpression of Rabl6A leads to larger tumors. And this is important because it reveals that the high levels of Rabl6A, which are seen in human patients, is tumor promoting. So based on that result, we wanted to test drugs that could inhibit Rabl6A signaling. But we don't have drugs that can actually inhibit Rabl6A. So therefore, we looked at drugs that target other factors downstream of Rabl6A. And we focused on reactivating a very powerful tumor suppressor called PP2A using small molecule activators of PP2A, or SMAP drugs. The exciting thing is that when we treated tumors with SMAP, shown in the blue curve, it was highly anti-tumorigenic compared to single drugs or the vehicle control shown in the black curve. So now let's consider three-dimensional cultures, models that we derive from patient tumors that are called spheroids and organoids. These are really useful because they preserve the original tumor biology, as they contain many different cell types that make up the patient tumor. A key first step is to verify that the organoid actually mimics and has the same features as the primary patient tumor. And here you can see that there are very similar staining patterns for synaptophysin, the Ki67 proliferation marker, and insulin between both the organoid and the primary tumor. So my colleagues at the University of Iowa, Drs. Po-Hin Ear and her mentor Jim Howe, have been generating patient-derived spheroids from patients for drug testing. So after Jim performs surgery on the patients, they isolate the tumors, and the team generates spheroids from small bowel and pancreatic neuroendocrine tumors and neuroendocrine carcinomas. They then subject these tumor cultures to treatment with over 175 different FDA-approved drugs. And they have found through those studies that they see differential responses with these patients. So on the left-hand side, each row is a culture from a different patient. And we have small bowel neuroendocrine tumor patients, pancreatic neuroendocrine tumor, and neuroendocrine carcinoma. And you can see a lot more blue in this heat map for pancreatic neuroendocrine tumors and NECs. And that's indicative of tumors that are responding really well to drugs. Now, by comparison, when you look at small bowel neuroendocrine tumors, you see a lot more white and red. And that indicates that these cultures are a lot more resistant to treatment with all of these different drugs that are shown up at the top that I know you can't read the names of. But basically, each column is a different drug. So the ultimate goal of this work is, after surgery, quickly generate these spheroid or organoid cultures, quickly screen each different drug for cytotoxicity using high throughput analyses, so that in a couple of weeks, they know which drugs could be useful against that patient tumor. Then they can use that information to guide patient treatment. Okay, so now we're going to move into the more complex system of in vivo testing using mouse models. And I've already told you about one type of model that is commonly used, in which human tumor cells are injected into immune deficient mice. Now, the limitations of this model are that the tumor cells basically will not go into the right place. So we have pancreatic neuroendocrine tumors, but we're injecting them into the flank, for example. Also, the mice are immune deficient, so they don't have an immune system. And otherwise, if they did, they would reject the human tumor cells that we're putting into them. On the other hand, these mice are great for early testing of putative cancer genes and potential drugs for therapy. Now, genetically engineered mice are really helpful because they mimic tumor mutations that are seen in the patient tumors. And over time, you'll develop tumors that arise in the appropriate tissue site, like the pancreas. Also, the mice are immune competent, like patients. And while this model may be expensive, it is definitely superior for preclinical drug testing and analyses of immune tumor biology. So the bioluminescent PNET model that I'm showing you here is really useful for testing if genes or drugs control pancreatic neuroendocrine tumor metastasis. So here we performed tail vein injections of cells that express a luciferase gene. And we imaged the tumor growth over a 7-week time period. And you can see with increasing luminescence that the tumors are growing. We then imaged the tissue distribution, and we see that some of these cells go into many different tissue sites in the animals, and that the QGP1 PNET cell preferentially homes to the liver, which is very interesting and relevant for human patients because that's where PNET tumors often metastasize to. Now, we've used this model to test drug combinations at how well they would inhibit tumor metastasis. And in our case, we're testing inhibitors of tumor-promoting kinases called CDK4/6 and MEK. And the representative images of mice shown below indicate that while tumors grow very well in vehicle control treated animals, the combination pretty much wipes out the tumor formation. Moreover, the combination also results in a reduction in the number of organ sites that are colonized by these metastatic tumors. So the mouse PNET models shown so far were immune deficient. That led us to collaborate with doctors Yuan, Latime, and Labudi at Rutgers to evaluate our drug combination in MEN1 PTEN double knockout mice, which are a model of insulinoma, and they are immune competent. These animals were of interest to us because they express high levels of our target CDK4. And we expected to see better efficacy of our combination, including tumor regression, especially with the combination, in this model when using these drugs. So as predicted, the combination acted synergistically to reduce tumor size compared to each drug alone, as quantified in this graph, where the red bar is denoting the combination, and it's much smaller than either of the individual drugs alone or the vehicle controls, which grew to be very large. Now, this robust level of tumor regression suggests that we actually had induction of anti-tumor immunity. Therefore, we quantified the tumor-infiltrating immune cells in the vehicle control versus the combination-treated tumors. And we found that there was significant increase in the levels of B cells and plasma cells in the combination-treated tumors, as well as antigen-presenting dendritic cells. Now, those results were really exciting because B and plasma cells in other human tumors are correlative with improved patient survival. They also correlate with increased formation of immune cell aggregates shown in this picture, where we have T cells and dendritic cells in addition to the plasma cells and B cells. And these are very important structures or tertiary lymphoid structures that mount an immune response and are able to kill the tumors. Now, plasma cells are also associated with a patient's improved response to immune checkpoint inhibitor therapy using either anti-PD-1 or anti-PD-L1 antibodies. And if you haven't thought about immune checkpoint inhibitor therapy and how it works, this is a very basic tutorial. So we know that cancer cells express a protein called programmed death ligand 1 or PD-L1. That protein interacts with a protein called PD-1 on the T cell, your immune cell. Now, when we use PD-1 or PD-L1 antibody therapy, that blocks this interaction and it prevents the cancer cells from inhibiting your immune response. Now, your immune T cells can go about killing the tumor cells. And this led to our hypothesis that inhibition of CDK4 and or MEK kinases would sensitize or make PNETs become responsive to anti-PD-L1 immunotherapy. And we examined this again in the MEN1 PTEN double knockout mouse. And in fact, we found that the combination shown in blue of palbociclib, which targets CDK4, and PD-L1 antibody is very effective at reducing insulin secretion over the 4-week treatment period compared to either palbociclib alone, shown in gray, or PD-L1 antibodies, shown in red. And this correlated with a tremendous decrease in the size of the pancreatic neuroendocrine tumors. And this is tremendously exciting. So what we have found is that we can target two oncogenic kinases, CDK4 and MEK, to effectively suppress PNETs. This inhibition will lead to the induction of an immune response characterized by infiltration of B and plasma cells within the tumors. Also, CDK4 inhibition alone was found to make the PNETs responsive to immunotherapy. Now, this is compelling because patients are normally not responsive to CDK inhibitors alone or immune checkpoint inhibitors alone. So we are recommending that the combination should be used. So we're doing a lot of future studies in the laboratory, and I've listed a bunch of them here that will better characterize and understand how the drugs are working to affect your immune response. And with that, I want to thank my wonderful colleagues, not only at the University of Iowa as part of our neuroendocrine tumor SPORE, but my collaborators at Rutgers, as well as Michigan and North Carolina, and especially the folks who work in my lab. Without them, none of this work could be done. Thank you.